collection and examination of blood samples in sheep and goats. Generally, what we are doing, or the why we are doing the blood samples, the reasons are over here. Because the sheep and goats are diseases in sheep and goats are economic importance for the farmers. Then there is a loss of some money of the farmers due to the use of some unwanted medications. Then there is a losses of the sheep and goat due to the mortality because of the undiagnosed disease conditions. Then there are subclinical infections of the hemoprotozoal diseases, which may lead to the decreased body scores, and whatever the meat which has been developed from such animals may be not of that much of good nutritional quality. So for that purpose, we need to go for the blood sample examinations or the different clinical or laboratory investigations of the different samples. Overview of the today's topic is the indications, method of collections, method of preservation, method of transportation, processing of the blood samples, examination of blood samples, and ultimately, after examination, what interpretation and results we are getting. Accordingly, we have to go further and treat the cases and prevent the disease, either transmission or the we have to treat that different cases of the disease conditions. Now, indications, we can see indications in detail. Generally, routine health checkup in larger farmers, larger sheep and goat farms, we have to carry out the routine health checkup. It is a part of preventive health management. Then if the animals are wasting, weakness is there, anemia is there, we have to go for the blood sample examination. If there is a fever, anorexia, melena, repeat bleeding is there, anistus is there, recurrent mastitis is there, abortions are there, orchitis or swollen joints in male, in general in case of males. If these conditions are present in the farm, in that case, we should go for the blood sample examination, and we have to carry out the different blood test for the diagnosis of the particular disease conditions. Again, whenever there is a red urine, yellow urine, or Downer's sheep and goat syndrome, and generally during the outbreaks like FMD, HS, Leptospira, PPR, pox, blue tongue, we have to take the blood samples at the height of temperature to isolate some viruses or some bacteria. So these are the indications of the blood sample examination. Like this, red urine is there, anemia is there. So these are the things which can be or the indications in that we have to collect the blood samples. Now start with the routine health checkup in as a part of preventive health management. Age-wise, those farmers are very curious. They can go for this. So either some representative samples from lambs or kids, then from the growing animals, pregnant animals, milking animals. So likewise, they can collect the samples and find out the different profiles like glucose, hemoglobin, total proteins, calcium, phosphorus, like that. So that is a part of routine health checkup of that particular farm. Then CBC also one of the tool, we can say, complete blood count is one of the tool for the routine health checkup. Then the total protein and glucose. So these are the things which can be done for the routine health checkup in the big farms. Now, another indication, wasting, weakness and anemia. In spite of good feeding management practices, if the animals are showing symptoms of weakness and wasting, then we have to go for the blood sample examination. In that case, subclinical hemoprotozoal infections are highly suspected in such conditions. Wasting is there, weakness is there, anemia is there. So these conditions may go towards the subclinical type of hemoprotozoal infections. In that case, we have to collect the samples and go to the laboratory. So like this, anemia, weakness. Then another conditions, it is very commonly observed that fever. Whenever there is a fever, recurrent fever is there and which is not responding to the regular treatment or NSAIDs, in that case, hemoprotozoal infections are suspected. Then bacterial infections, viral infections, transportation stress and fever, like the shipping fever, generally because of the HS or the hemorrhagic septicemia. In that case also, we have to collect the blood samples and go for the diagnosis. Then anorexia, non-specific anorexia, anorexia associated with fever, non-associated with fever and transportation stress, anorexia. So in that case also, we have to collect the samples and go for the diagnosis. Melena. Now, melena is the black discoloration of the stools of the fecal samples. Melena associated with fever, then it is suspected for the hemoprotozoa, because in case of the thylaria, there is a formation of some ulcerations in the abomasum of the animals, and that lead to the melena. Melena associated with fever and enlarged superficial lymph nodes. It is 100% suspected for the theileria. Melena not associated with fever and enlargement of lymph nodes. Possibility there are the chances of the overdoses of the non-steroidal anti-inflammatory drugs. So can't be differentiated based on the blood examination. So these are the things we should keep in mind. So like this, melena will be there. Repeat breeding. Now this is a very common problem in many of the farms. One of the common conditions associated with the subclinical hemoptyosal infection is the repeat breeding. Then deficiency anemia, deficiency of minerals could be possible, that may lead to the repeat breeding. Deficiency of vitamins and proteins, that may also lead to the repeat breeding. Then high level of blood urea nitrogen. This is very commonly observed in case of the high or the heavy builders, heavy breeds like boar. Due to the high protein diet, most of their owners, they are giving the high protein diet, that lead to the increase in blood urea nitrogen levels, and that lead to the repeat breeding or the unstress conditions in case of the heavy breeders. Unstress, again the same thing. Subclinical hemoptyosal diseases may lead to the unstress. Then there are chances of the deficiency of nutrients. Then hormonal disturbances. So most of the times leptin overexpression may lead to the unstress phase in the sheep and goat. Then there is a decrease in FSH and LH, that is very commonly observed in the heavy breeds. Then the low and high cholesterol levels. So low cholesterol levels in the lean breeds, and that is associated with the malnutrition, and high cholesterol levels is associated with the or the high cholesterol levels and unstress is associated with the heavy breeds. Recurrent mastitis. Again, the same thing is applicable over here. Chronic hemoptyosal diseases, then the chronic liver dysfunction, improper assimilation of nutrients, low albumin levels, low globulin levels, and because of that, there is a low metalloproteins, which plays very important role in the immunity boosting of the mammary gland. Then this hemoptyosal infections, subclinical hemoptyosal infections may lead to the recurrent mastitis in case of sheep and goat, and deficiency of minerals, especially calcium. So all these conditions may lead to the recurrent mastitis. So whenever there are problems of the recurrent mastitis, instead of going only milk sample examination, we should go milk plus blood. So we will get the complete diagnosis of that particular case. Then abortions in females, orchitis in male. Generally, it is giving us or giving some clues about the brucellosis. So in case of brucellosis, these are the clinical signs: abortions in the females, and there may be the orchitis and swelling of the joints is observed in case of brucellosis. Brucella abortus may be there, melitensis may be there. Now red urine, whenever there is a red urine, again we have to go for the blood sample examination. So that may be because of the babesia, hypophosphatemia, leptospirosis, and sometimes water intoxication. If the growing animals consume more water, in that case also there is a change in the colloidal osmotic pressure and RBC get rupture, and that may lead to the red urine in case of sheep and goat. Then clostridial infection like Clostridium hemolyticum also lead to the red urine. So in that case also we have to go for the blood sample examination, and along with that we should go for the urine sample examination too. Yellow urine, so it is also indication of the hemoptyosal diseases, area, anaplasma, mycoplasma, liver dysfunction, obstructive jaundice, like the conditions like dehydration, severe dehydration. In that case also we should get yellow urine. So these are the some indications for the yellow urine. Now Downer's syndrome in sheep and goat. So here we should have the some clinical correlations with the Downer syndrome, and what are the different clinical signs. So generally many people trying whenever the sheep and goat get sit down or the show the symptoms of Downers, generally they suspect that there is a deficiency of calcium, phosphorus, potassium, and sodium, like that. But beyond that, so whenever there is a DIC, means a disseminated intravascular coagulopathy, because of the hypercoagulation in the bloodstream due to the extensive number of platelets, that may lead to the Downer sheep and goat syndrome. Then acute mastitis also lead to this due to the severe pain. Sheep and goat may sit down, and we can call as a Downer syndrome. Then acute muscular injury, if the sheep or goat fallen on the ground or maybe get bite by some other sheep and goat, in that case also due to the severe pain, sheep and goat may be sit down and we may call as a Downer syndrome. Then dystocia, sometimes the fetus is so big, so in that case there may be the chances of the injury to the spinal cord, and that may lead to the Downer syndrome in case of sheep and goat. So we have to correlate everything and don't jump directly on the deficiency of the calcium and phosphorus. So before that, we must have the exact clinical correlation with that particular case. Now, the other thing is that the seroprevalence during the outbreaks of the some of the disease conditions like the FMD, HS, leptospira, PPR, pox, blue tongue, etc. We have to collect the blood samples. Then we have to collect the blood samples for the post-vaccine antibody titers. So many times vaccines has to be given to the sheep and goat, and to monitor the is there is a good efficacy of that vaccine or not, we have to collect the blood samples, serum samples, and go for the antibody titers. So that will be called as a post-vaccine antibody titers. And this is to be seen for the efficacy of the vaccine. So these are the indications of the blood sample examinations. Now we will shift towards the collection of blood samples. So generally, the site of collections, it's the most commonly used the site is a jugular vein, most common site. Ear vein, sometimes in case of the some non-cooperative animals also we can go for the ear vein. Then femoral vein, recurrent tarsal vein, but it is rarely used in the sheep and goats. Ear tip is the most commonly used to detect the hemoprotozoal infections. Peripheral blood smear examination we can call it. Should be collected after clipping the hairs and cleaning the ear pinna. Many times people are puncturing the ear vein and consider as this is the peripheral blood smear, but that is wrong. We should take a blood smear from the tip of ear, and that will be considered as the PBS or the peripheral blood smear. It is not the puncture of the ear vein. So like this, this is a collection of blood samples. Usually everybody knows how to collect the samples. So it's a jugular vein. Jugular vein, we have to raise the veins and insert the needle and collect the blood samples. So it's a very easiest method to collect the blood samples. Then we have to transfer it into the EDTA tubes and we have to mix it very well. So whatever the powder or the solution which is present in that particular tube, we have to mix it carefully. So the powder of the EDTA is get mixed with that particular blood sample. So it may not get clot. So this is a collection of blood sample. Again, another case of the same thing. Now, how to take from the tip of the ear. So this is a dirty ear pinna. We have to clean this ear pinna. Either we can go for the washing of this, then clean it and take the blood smear from this by just pricking the needle over here. So at the tip of this ear pinna, we have to prick the needle and take a small drop from this and make a smear of that. So ultimately, this is the peripheral blood smear examination. Now, the blood and its different tests. So after collecting the bloods, what we have to do or what has to be done in the laboratory. So the first test is very commonly used is the CBC, the complete blood count. So here we need the whole blood and it should be in the heparin or EDTA. Now it should be kept below 8 degrees centigrade. So then the next is a hemoprotozoal disease. In that case also we can collect the whole blood in EDTA or heparin and we should keep the vial below 8 degrees centigrades. For the hemoprotozoal disease, just if you want to take the peripheral blood smear, then we have to take the one drop from the tip of the ear and make a smear, ear dry it or fix it with the methanol, wrap in the paper and send for the diagnosis or the you can on your own you can stain it and identify the for different conditions. Then liver function test, for that purpose we require the serum. Then Preservative, no any preservative should be added in the serum for the liver and kidney function test, and it should be carried in the ice only. For electrolyte also, either serum or whole blood or plasma can be used for the electrolyte analysis. So here we can use the heparin, and it should the sample should be transported through ice only. Then serology of the brucella, FMD, blue tongue, tuberculosis, everything we should require the serum, no preservative, and it should be carried on ice only. Now again the other test, like the test for the bacterial or the viral isolation, we have to collect the samples at the height of temperature. Whole blood is required in a sterile container. It could be EDTA, heparin, or sodium citrate. It should be transported on the ice only. Then serum for mineral profile, we should require serum. No preservative should be added, and it should be transported through ice only. Blood glucose, whole blood required, but it is in the sodium sulfate only, and it should be transported through ice only. Protein and lipid profile, serum will be required. No any preservative will be required and should be transported through ice. Blood compatibility, whenever severe anemic cases are there, and in that case we have to go for the transfusion. In that case, we may require the donors and recipient blood for the compatibility testing. So it should be transported on ice and with the sodium citrate. Then blood fibrinogen level, whole blood is required, and it should be in the heparin and should be transported through ice. So blood fibrinogen level is generally required in case of the snake bite cases. Now these are the tubes. So here you can, if you see carefully, the K3 EDTA. So these are the EDTA tubes which can be used for the CBC, blood hemoprotozoal diseases. Then this is also same tube. Then for serum tubes, we should require the clot activator tubes or plain tube. So here you can see the name has been given, the clot activator. So due to the clot activator, the RBC parts get separated and the clear serum will get separated. We can transfer it into the another container, or you can send directly this type of container to the laboratory for the investigations. Labeling is another important part of the sending the samples to the laboratory. Here we should provide name of the farmers, age, sex, breed, it should be there on the bottle. Then purpose of blood samples examination. If anybody wants to go for the CBC, serology, serum biochemistry, etc., it should be mentioned on the particular blood vials. Avoid direct contact of ice to the bottle, because whatever the labels we have mentioned over the bottle, it may get spoiled. So the better thing is that we should keep the all vials into the plastic or polythene bag, and then that bag should be transported, or the transport to the ice, or the around that bag we should keep ice or ice packs. Do not freeze the whole blood. So generally what happens, if the samples has to be sent on the next day, many people collect the samples and keep the samples in the deep freezer. So because of that, in deep freezer, animals the samples get hemolyzed and it will not have any use, and it get hemolyzed. So no any test can be done, no hemoprotozoa, no serum, or nothing can be done with the samples which has been freeze. Whole blood, I am saying about the whole blood. Whole blood should not be freeze anyways in the freeze. It should be kept at the lower side of the freeze. So below 8 degrees centigrade, wherever you are putting the all vegetables, we can put that samples in that place only. Serum, but the serum can be put at the freezer. So if you are sending samples very late, then you can put the serum, but it should be separated serum. It's not the clot activated tubes directly put into the freezer. So we have to separate the serum and then put into the deep freezer. Then you can transport accordingly as per your convenience. Now, the another factor or the part of the blood sample examination is the processing of blood sample for CBC, though complete blood count. After receiving samples to the laboratory, mix the samples very gently. Do not shake the samples to avoid vigorous mixing. So if we go for the vigorous mixing, definitely some of the RBCs get ruptured, some of the cells get ruptured. So don't go for the vigorous type of mixing. But after that, after gentle mixing of the samples, prepare the blood smear, allow it to air dry, then label the blood smear and then go for the staining. Then the other test like the packed cell volume, we have to fill up the Winthrop tubes, then fill up the ESR tubes to see the ESR, then carry out the hemoglobin, prepare the sample for the RBC, and for that purpose we are going to use the Newbar chamber for RBC and WBC count. So these are the routine things can be done in the laboratory. Now this is the blood smear. Blood smear examination, you can see the if n numbers of samples are there, we can give labeling to the each and every slide. So simple method is the just with the pencil, we can put the number of that particular animal. Whatever the labelings are there, we have to put over the blood smear. It won't get washed in the staining. So if we are making mark with the marker, so marker may get spoiled and it get vanished due to the Leishman stains or the other stains, but the pencil marks will be remain as it is forever. And after staining also, these marks will be remain as it is. So here, so once the slides get dry, we should go for the staining of the slides. So like this, put the stain, allow it for the 1 to 2 minutes based on the concentration of the our staining solutions. So this is a Leishman stain. So after 1 minute, 1 to 2 minutes, again you have to give. We have to put the water. So like this. We can put the water and allow it to react for the 10 minutes. After 10 minutes, our slides will be ready, and you can see the whatever markings we have made over the slides has been remain as it is. So nothing has been happened to that marks. So the whatever the numbers are there, 1, 2, 3, 4, 5, and these are the hemoglobin levels has been kept over the that slide. So it is remain as it is. So there is no change in the labeling. So here, if you see the platelet count in that particular goat, it's too huge. It is double than the normal size, normal number of RBC. So all these are the platelets over here. You can see the number of RBCs are less, but the number of platelets are so heavy. So these are the some of the conditions that can be observed in the field conditions. So examination of blood smear samples. So stain blood smear examination is the most important part of the diagnosis in sheep and goat. No any other options for the microscopic examinations. Many times though, so many machines are available, but there is no any alterations for the identification of the some of the abnormalities, then the changes in the blood smears. So we have to go and observe the smears. That is the only way. The focus the blood smear first on the 10x, then go for the oil immersion or 100x. Observe the smears in a zigzag manner. Observe the morphology of RBC, WBC, and platelets. Count the 100 cells and complete the differential count. So DLC should be completed by counting the 100 cells. Then we should go for the specific conclusions of the particular blood samples. Like this zigzag manner. So we can start from this like this. Again, just shift the one focus, go to here, and again the like a zigzag manner we have to examine the slides. So from top to bottom, like that, or bottom to top to bottom, like that. We have to go for the blood smear examination. Now, many times the tail of blood smear is not examined by the many peoples, but it is a most important factor that the tail of the blood smear is a very important factor or the important part of the blood smear to be examined. So in the tail of the smear, different type of cells are there, and the morphology because in the tail portion there is a density of the cell is very less. So that's why we are able to identify hemoprotozoa very clearly, then the platelet morphology very clearly, and the morphology of the other cells also very clearly. So don't forget to examine the tail portion of the blood smear. Then processing of the serum sample. So after examination of the blood smear, we must know what to do with the serum. Serum samples are to to be processed as per the requirements, like the liver function test. For that, ready-made kits are available. Then kidney function tests also, the ready-made kits are available. Serum calcium, phosphorus, magnesium, potassium. Here also ready-made kits are available. Then the another factor is the brucella. So for brucella, we can go either for the RBPT, ELISA, or the STAT. The ELISA and STAT. These are the very commonly used. RBPT also one of the tests can be used for the screening of the different flocks. So for the brucella serology, we are having the options of RBPT, ELISA, and STAT. Then serology for the different disease conditions like ELISA for TB, JD, PPR, blue tongue, and FMD. So these are the disease conditions can be diagnosed with the serum sample. Then interpretation of the results based on the hematological findings. So we should go for the interpretations and what conclusions we are getting. Just we have to discuss that, and whatever the biochemical parameters we are doing in the laboratory, and what are the different normal values and how to go for the conclusion. We will see that particular portion. Now here it's a simple things. So normal values of hemoglobin, RBC, WBC, then platelet, then PCV, MCV, MCH, and MCHC. So these are normal values in sheep and goats. So accordingly, any change in that particular values, we can correlate the different disease conditions. Normal values of the hematology, so especially the differential count, neutrophils, lymphocyte, monocyte, eosinophil, basophils. So these are the generally observations or comparisons we must or these value we should have with us for the comparison of the whatever values we are getting from our diagnosis. Result and interpretation of hemoglobin. Now hemoglobin is 8 to 14, so we can say it is healthy. Below 8 to 6, 8 to 6 mild anemia, then 6 to 4 moderate anemia, less than 4 it's a severe anemia. Now the conditions are somewhat remain same. So 8 to 14 will be called as a healthy. Mild anemia may be due to the mild enteropathy, chronic hemoptyosal infections. Moderate anemia, it's acute hemoptyosal diseases, severe enteropathy, and blood loss and melena. Severe anemia, acute hemoptyosal disease, severe enteropathy, blood loss, and melena. Now, beyond that, some of the other conditions are there. If the hemoglobin concentration is more than 14%. So in that case, the higher values of hemoglobin is indication of the hemoconcentration. Maybe dehydration, maybe heat stroke, maybe diarrhea, and maybe sometimes due to the use of diuretics or the hyperpnea cases, or the whenever there is a respiratory distress, in that case also the hemoconcentration is there. Generally nowadays, whenever there is a dehydration due to the heat stroke, such type of conditions can be observed in the blood. Now, total leukocyte count, it is one of the crucial part of the disease diagnosis or the blood examination. So generally, total leukocyte count is 4 to 13. So it will be called as a healthy animals. If go beyond 13 to 18, mild leukocytosis, 18 to 25, moderate leukocytosis, and above 25,000, it will be severe leukocytosis. So these are the conditions which lead to the different alterations in the leukocytic count. 13 to 18, mild acute inflammation could be stress, any kind of stress. Moderate leukocytosis can be observed in the moderate inflammation, acute infections, and severe stress. Severe leukocytosis can be observed due to the many suppurative conditions, suppurative inflammations, injuries, mastitis, metritis, pneumonia. So in all these conditions, the total leukocytic count will be more than 25,000. Then if the count is less than 4,000, so generally leukocytopenia is observed. It is due to the immunosuppression and immunosuppressive drugs, viral infections, and bone marrow suppressions. So these are the conditions and interpretation of the TLC. Then the platelet count, it is one of the crucial things observed during the blood sample examination. The platelet count is a 3 to 7.5 is a normal range, 7.5 to 20. So mild thrombocytosis, 21 to 40 is moderate thrombocytosis, 40 to 40 and above, severe thrombocytosis. So these are the conditions. Again, the acute inflammation, vascular endothelial damage, and its intensity. If the mild inflammation is there, mild endothelial injury is there, and toxemia is there. So we are getting about 20 lakhs platelets per ml. And as the intensity goes beyond the capacity of the particular injury or the repair, then we are getting the moderate thrombocytosis, severe thrombocytosis, and thrombocytopenia. Whenever there is a less than 3 lakhs thrombocytes per ml, in that case, bone marrow suppression, rickettsial infection, and some of the toxicities are indicated. Then interpretation of the neutrophils. About generally 50% of the neutrophils are present in the sheep and goat. It could be up to the 15 healthy, we can say 50 to 60 mild neutrophilia, 60 to 70, we can say the moderate, and 70 and above severe neutrophilia. So the etiological factor will be remain almost same. Stress will be there, acute inflammation will be there, acute inflammation, use of steroids, bacterial infection, supportive inflammation. So all these conditions may lead to the increase in the neutrophilic count. And whenever there is a neutropenia, the neutrophilic count will be less than 10, and it could be observed in case of the hematopoietic infection, viral infection, and bone marrow suppression. Then lymphocytes, it should be 50 to 70 normal range, 70 to 80 mild lymphocytosis, 80 to 90 moderate, and 90 to above the severe lymphocytosis. So here, in contrast to the neutrophilia, we are getting the chronic inflammation, hematopoietic infection like anaplasma thalaria, moderate to chronic inflammation, and again the hematopoietic infections and thalaria. Severe chronic inflammation or the long-standing chronic inflammation and hematopoietic infections are indicate in case of the severe lymphocytosis. Whenever lymphocytic count is less, then there are chances of the steroids given to that particular animal, immunosuppression, and severe acute inflammation, acute bacterial infection. So in that case also, we are getting the lymphocytopenia. Now, eosinophils count, generally, many times eosinophil count get increased in case of the parasitic infestations and the allergic conditions, as well as the hypersensitivity reaction. So these are the some of the stages like normal, mild, moderate, and severe eosinophilia. Based on the etiological factor, there are changes in the eosinophilic count. Then the monocytes, generally chronic inflammation is indication of the monocytosis. Monocytosis can be observed in case of the tuberculosis, Jones disease, and listeria. And tuberculosis is very commonly observed, and Jones disease also very commonly observed in case of sheep and goat. Now we can see the different hematopoietic slides. So thalaria, babesia, anaplasma, and mycoplasma. These are the very commonly hematopoietic diseases can be observed in sheep and goat. Depending upon the intensity, we can classify as acute and subacute or subclinical, we can say. So anaplasma. So here we can see the two dots like structures. So this is the anaplasma marginal, central. Here also we can see the anaplasma marginal. Here also we can get the anaplasma central. So these are the anaplasma. Then thalaria, different shapes and size of the thalaria can be observed, like a comma shape, then the ring shape, again the ring shape. So these different type, this is also ring shape. So these are the different types of the thalaria can be observed in sheep and goat. Again, thalaria in in goat. Here you can see the number of RBCs are infected with the thalaria. Then KBB is one of the common finding. So you can see the coach blue bodies in the cytoplasm of the lymphocytes. Then babesia, different stages of babesia can be observed in the blood smear. So like two banana type of cells can be observed, pyroplasm we can say, observed in the RBCs. Here again babesia, it's a clump of babesia present over here, and this is two. Here one is one. So like that, see, this is babesia get propagate by binary fission in the RBC to RBC, and the infection get propagate, and ultimately the animal will show hemoglobinuria later on. Babesia in goats, again the same thing. You can see the number of babesia infected cells like this. Mycoplasma, it's again common conditions in sheep and goat. Here also you can see the mycoplasma. Sometimes there are confusion between the mycoplasma and anaplasma. So based on the its size and shape, we can differentiate, because anaplasma is a dot, round dot, but the mycoplasma will not have the round drop. It is a like a dish, or the we can say the saucer shape type of mycoplasma can be seen in the RBCs. Now, the next thing is the serum and its different concentration of the different parameters. So the total protein, albumin, globulin, SGPT, SGOT, alkaline phosphatase, bilirubin, creatinine, and blood urea nitrogen. So these are the very commonly used parameters in case of sheep and goats. Then minerals, calcium, phosphorus, and magnesium. These are so minerals can be used, or the minerals can be diagnosed in the laboratory. Now, the important factor is the important part of the interpretation of the serum values. So total protein, generally high protein cases are not observed, but low protein cases are observed commonly in field conditions. So low protein concentration can be observed in case of malnutrition, enteropathy like coccidiosis, malnutrition, then liver dysfunctions, poor diet, chronic exposure to the toxins. It is a very common conditions, mycotoxicosis. In that case also, we are getting the total protein count is less. Similarly, in albumin and globulin count will be seen less in case of the same kind of the conditions. Then higher globulin levels generally observed in case of the acute phase reaction. Whenever there is a acute inflammation, acute mastitis, fever, persistent inflammatory conditions, abscess is there, metritis is there. In all these conditions, we are getting the higher albumin and globulin ratio, and that is indication of some injury or the inflammatory reaction in the body. Then higher SGPT levels indicates the liver dysfunction, chronic venous conditions, and mycotoxicosis. Then SGPT will be higher in case of liver dysfunction and muscular damage. Alkaline phosphatase will be observed in case of the bile duct inflammation and injury. Then acute inflammation, any kind of acute inflammation, and bone reabsorption conditions. So in all these conditions, we are getting increased alkaline phosphatase. Then interpretation of the biochemicals for the kidney function test. So blood urea nitrogen generally find higher values in case of the intestinal and abomasal bleeding, renal impairment, and high protein diet. So whenever there is a high protein diet, generally observed in case of the heavy breeds, so blood urea nitrogen levels are generally high. Then creatinine levels are higher values, generally observed because of the renal impairment, chronic NSAID uses, then the multiple disseminated abscess in kidney, then mycotoxicosis and high TDS of the water. Whenever the TDS of water is high, in that case also there are chances of the increased creatinine levels in sheep and goat. Then lower calcium levels that can be observed because of the poor quality feedstuffs, high phosphate containing diets, vitamin D deficiency, more hardness of water, more TDS of the water, and high oxalate in feed. In that conditions, we are getting the low calcium levels. Then whenever there is a low phosphorus levels, it is again the same thing. Poor quality feedstuffs, vitamin D deficiency, more hardness of the water, more TDS of the water, and high oxalate content in the feed. All these conditions lead to the hypocalcemia and hypophosphatemia. The magnesium, whenever the pasture or the grasses are less in magnesium, we are getting the low magnesium values. Whenever there is a low whenever there is a high molybdenum in the feed, in that case also there are chances of the decrease in the magnesium availability for the pastures or the feed that we are growing in the particular land or the field. Then potassium, lower values maybe because of the dehydration, renal impairment, and poor quality feed. So these are the interpretation of the serological examinations or the serum biochemistry. Then serology for brucella, we can go for the RBPT, ELISA stat. Serology for different diseases like tuberculosis, JD, PPR, blue tongue, FMD. Reports will be positive or negative. So the animals may be positive for brucella or maybe negative for brucella. Now, in case of the sheep and goat, we should keep much more, here we can say I am giving the special note for the tuberculosis. Now, what happened in tuberculosis? This is a zoonotic disease, no doubt. But in case of up till now, whatever I observed in the those sheep and goat affected by tuberculosis, the common factor is the human beings. So infection from human is go to the sheep and goats. So humans working in the farm should be tested once in a year for the tuberculosis, as these labors will be the spreading factor for, of the tuberculosis in the farm. I want to give special note for the tuberculosis because I have seen many cases of the tuberculosis in sheep and goat. And whenever I go to the farm and observe the farms, then it was noticed that the workers which are present in the farm, they are generally, as we know, they are weak, emaciated, and they are having the tendency to continuously cough, maybe men or women, and they are spitting here and there in the farm, and ultimately they can contaminate the surrounding, the flies whichever present over there, they can contaminate feed and water, and likewise this tuberculosis gets spread, and ultimately we have to kill all the animals if the animals are having the tuberculosis. Now we are going towards the last part of our today's webinar. So blood sample examination summary. So it is very important at the field conditions. So blood smear examination and CBC estimation is a very common, very important as a tool for the diagnosis of the different conditions. We should go by the proper method of collection of blood sample examination. We should go for the proper method of transportation of the and the transportation medium for the blood sample examination. And there is no any option for the microscopic examination of the for the hemoprotein diseases. Then the cellular morphology and cellular abnormalities. Then you have to strengthen your diagnostic approach by using the blood sample examination. Plan, and after that we have to plan the different preventive strategies for the management of the spread of the disease infections or disease conditions. Then comprehensive approach in the diagnosis of the laboratory diagnosis of the blood smear or the blood samples is most important. Observation of the surrounding is most important factor in the farm, like how the feed samples has been stored, how the cleaning of the farm is to be carried out, how is the biosecurity measures in that particular farm, and we must know the types of water in the farms, must know the types of people working in the farm, must know the how they are going to dispose the biological waste material in that particular farms, like the dead fetus, fetal membranes, infected milks, like that. So that will be the source of infection, and ultimately other animals may get infected by that biological material. And see further where the farmers are selling the animals for the brucellosis. Generally, if they are selling to the other farms, then definitely that farms also get affected by the brucellosis. So these things should be we should keep in mind for the brucellosis. So here again, the understanding the pathology of the suckling. Most of the times the people know that there is a whenever there is a theileria, there must be the presence of high fever. So ultimately, if the animals are not showing the fever, then the conditions may be unnoticed, and the animal become a so slowly weak, and finally died because of the anemia. So in case of the subclinical hemoprotozoal infection like theileria, anaplasma, babesia, and mycoplasma, there is a severe problem in the field conditions, and most of the times these type of cases are get ignored by our regular clinicians. So because these animals does not show any fever, there is animals will show intermittent coughing, recurrent illness, recurrent mastitis, and maybe get cure by the some routine treatment, but the subclinical infection remain as it is in that particular animals. Animal will show the wasting, animal will show the anistrus. So whenever animals show anistrus in spite of good mineral, good quality of the feed, then we should suspect that cases for the chronic hemoprotozoal or the subclinical hemoprotozoal infections. Now clinical correlation of the laboratory diagnosis is most important, and it will enhance the recovery and minimize the losses of the farmers. So these are the things we should keep in mind while doing the practice in the field conditions. So these things can be from my side.